Alright, welcome everyone. This is my third lecture that I'm going to upload, and it's the continuation of the chapter Cell Cycle and Cell Division. And the topic for today is meiosis, which will also mark the end of this chapter. And yeah, so my name is Ojasvi Singh, and these are some of my qualifications. Uh, Before I begin the lesson, I would like you all to like, share, and subscribe because my incentive out of all of this is that I want to reach as many people as possible because there would be no point in posting content if there are no people to watch your content and to get benefited from it. So I want to reach as many people as possible so that I can benefit as many people as possible. So yeah, that's pretty much it. Kindly share the video with your classmates and anyone whom you know who is preparing for NEET, because I'll be giving a lot of insights which might be useful, right? So yeah, and in today's lesson we'll only be talking about meiosis. We'll complete it, and in the next lesson that I upload, we'll be discussing the last 10-year questions that have come from this chapter. All right, so let's move. Yeah, so let's just go through the topic, the lines one by one, because NCERT is the gold standard, and everything will be coming from the book. And if there are some extra things that might come in the exam, I'll also let you know that so that you can make proper notes of it. Alright, so let's dive into meiosis. Meiosis is basically the production of offspring by sexual reproduction, includes the fusion of two gametes. So for the fusion of two gametes, you first need to have two gametes. So meiosis is the process which causes the formation of gametes. Okay, each with a complete haploid set of chromosomes. Gametes are formed from specialized diploid cells. So initially there is a diploid cell, and after meiosis you will get haploid cells. Okay. This special kind of cell division that reduces the chromosome number by half results in the production of haploid daughter cells. This kind of division is known as meiosis. Meiosis ensures the production of haploid phase in the life cycle of sexually reproducing organisms, whereas fertilization restores the diploid phase. Right. So this is your diploid state. After meiosis, in which a lot of different processes occurs like recombination, etc. So after meiosis you will be getting haploid cells. This haploid cell will fertilize with another haploid cell, and eventually the organism, that is the child that will be born, will be having diploid chromosome number. Alright, so we come across meiosis during gametogenesis in plants and animals. This leads to the formation of haploid gametes, and the key features of meiosis are that meiosis involves two stages. Two stages, one is meiosis 1 and the other one is meiosis 2, where meiosis 2 represents a pattern similar to mitosis, but only a single cycle of DNA replication, right? Because initially the first stage is your replication of the DNA content, right? So if your number of chromosomes is 2n and the number of the genetic content is also 2n, then in the synthesis stage that occurs before even M1, in the synthesis stage you will get chromosome number as 2n, but the DNA content becomes 4n, right? So this S stage occurs, so this is the pattern. The S stage, after S stage will come meiosis 1 and then will come meiosis 2. Okay, so the S stage occurs only one. That means that DNA replication occurs only once. DNA replication occurs only once. Alright, then meiosis is initiated after the parental chromosomes have replicated to produce identical sister chromatids at the S phase. So this is the same thing. Now since the DNA has become 4n, initially this was the structure of the chromosomes. Now the chromosomes become this because of replication of the DNA. So this was your first copy. After replication, this is your second copy, and these two chromatids are known as your sister chromatids, because they are identical to each other, sister chromatids. And this also occurs in the S phase because your genetic duplication occurs at S phase. So meiosis 1 is initiated after the parental chromosomes have replicated to produce identical sister chromatids at the S phase. Meiosis involves pairing of homologous chromosomes and recombination between them. So this is your first set of chromosome, this is your second set of chromosome, right? Pairing of homologous chromosomes. Now these are known as homologous chromosomes. These two are sisters, so sister chromatids. And these two are non-sister chromatids because they are on different chromosomes, right? So non-sister chromatids. Between the non-sister chromatids, recombination occurs. Alright, so we'll look more into it as we proceed in the lesson. Okay, so 4 haploid cells are formed at the end of meiosis 2. Meiotic events can be grouped into meiosis 1 and meiosis 2. Now meiosis 1 has prophase, metaphase, anaphase, telophase, like the stages of mitosis: pro, meta, ana and telo. Pro, meta, ana and telo. Okay, but in the prophase 1 of meiosis, again it is divided into multiple different stages, like we'll see in the next slide what are the different stages of prophase. So let's move ahead. Yes. So when talking about meiosis 1, the beginning is prophase. Of course, the beginning is prophase 1, pro, meta, ana, telo. But the prophase 1 is also divided into subcategories. So prophase of the first meiotic division is typically longer. Of course, it has so many stages, so it becomes longer and more complex when compared to the prophase of mitosis. It has been further subdivided into the following 5 phases, namely lepto, zygo, pachy, diplo, and dia. So first let's write down each stage, and this is how I remember also, that lepto, zygo, pachy, diplo, dia. Lepto, zygo, pachy, diplo, dia. So you can memorize it in that same manner. This is pretty much the only memorization that you will be needing in this lesson, because the rest of the things follow a story. So if you remember the story, then there's no need to memorize what happens there. Okay. So let's start with lepto. Leptotene, lepto, zygo, pachy, diplo, and dia. So during leptotene stage, the chromosomes become gradually visible. So visibility happens in leptotene. The compaction of chromosomes continues throughout leptotene. This is followed by the second stage known as zygotene. So in the leptotene stage, what happens? Chromosomes become visible. Let me just write it down completely. Chromosome becomes visible due to the chromosomes becoming compact. Alright. Then this is followed by the second stage of prophase 1 called zygotene. During this stage, chromosomes start pairing together. Okay, so over here what has happened? The chromosomes have become visible. But in the zygotene stage, the chromosomes start pairing up. They start pairing. Start pairing. Uh, this is followed by second stage called zygotene. During this stage, chromosomes start pairing, and this process of association is called synapsis. So this is known as your process of association is known as your synapsis. Such paired chromosomes are called homologous chromosomes, synapsis, and homologous chromosomes. Electron micrographs of this stage indicate that chromosome synapsis is accompanied by the formation of a complex structure known as synaptonymal complex. The complex formed by a pair of synapsed homologous chromosomes is known as bivalent. So this pair of chromosomes is also known as bivalent, okay, or a tetrad, bivalent slash tetrad. These are the words that have come in the past 10 years of your questions, and they are likely to come again. So whenever a question comes which says which is the stage of meiosis in which tetrad formation occurs, you have to go with zygote. So that is how you have to connect that what happens at which stage. Okay, so if the question comes like the stage of meiosis in which the chromosomes become visible, then you will go with leptotin, right? However, they are more clearly visible at the next stage. The first two stages of prophase 1 are relatively short-lived compared to the next stage, that is the Pagite. Now let's move to Pagite. During this stage, bivalent chromosomes now clearly appear as tetrads. Okay. So in the Pagite, there is a clear appearance of tetrad. This stage is characterized by the appearance of recombination nodules. So recombination nodules become visible at this stage. So what are recombination nodules? Basically what happens if this is one pair of sister chromosomes, chromatids, and this is the other pair of sister chromatids. Over here, recombination is occurring. So this recombination occurs inside the recombination nodules when the homologous chromosomes are arranged in this pattern in the synaptonemal complex in a bivalent. So over here your recombination is occurring. This all happens in your Pagite stage. Okay. Then the sites at which crossing over occurs between non-sister chromatids. Crossing over is the exchange of genetic material. So this is the definition of crossing over. It will not come in neat, but it will come in your school exams and in your 12th boards. Alright, so crossing over is the exchange of genetic material between two homologous chromosomes. Crossing over is an enzyme mediated process, and the enzyme involved is known as recombinase. It has not come in neat as such, but it is important. It may come, and it will obviously come in your other entrance examinations like CUET, etc. So, crossing over leads to recombination of genetic material on the two chromosomes. Recombination between homologous chromosomes is completed by the end of Pagite. So, by the end of Pagite, recombination is completed, leaving the chromosomes linked at the sites of crossing over. However, even though the recombination is completed, this linkage is still present at the end of Pagite. Let's move to diplotin and diakinesis. So the beginning of diplotin is recognized by the dissolution of the synaptonemal complex and the tendency of the recombined homologous chromosomes to separate from each other except at the sites of crossover. The X-shaped structures are called as chiasmata. So what happens is that the chromosomes now start separating from each other. However, this part is still not separated, so synaptonemal and the tendency of the recombinant homologous chromosomes of bivalence to separate from each other except at the sites of crossovers. The X-shaped structure are called chiasmata. Sometimes the recombination may also occur like this. I'll just draw this. At the top also is recombining, at the bottom also is recombining. So chiasmata formation occurs at diplotin. When you're talking about diplotin and its marker event, the marker event is formation of chiasmata. Okay. So if a question comes which says which is the stage in which chiasmata is formed, then it would be diplotin. In oocytes of some vertebrates, diplotin can last for months or years. This question has come in neat. I don't remember the year, but it has come, and it is a very important statement. You will know its significance when you will be studying reproduction in animals. So the oocytes of some vertebrates, diplotin can last for months or years. So the question comes like this: which is the stage in which the oocytes are arrested for months or years? So it would be your diplotin stage. Okay. The final stage of meiotic prophase 1 is diakinesis. It is marked by terminalization of chiasmata. So when we are talking about diakinesis, its marker event is terminalization of chiasmata. During this phase, the chromosomes are fully condensed and the meiotic spindle is assembled to prepare the homologous chromosomes for separation. By the end of diakinesis, the nucleolus disappears. This is again one of the characteristics. Nucleolus disappears. Nucleolus disappears. And the nuclear envelope also breaks down. Diakinesis represents transition to metaphase. Nucleolus and nuclear membrane. Let's write both. Nuclear membrane and nucleolus disappears. Now let's move to the next stage, which is metaphase 1. Till now we have only been studying about the prophase. As I said, it is the longest stage of your meiosis 1. So now let's move to metaphase. So in metaphase, the bivalent chromosomes, till now what has happened is in your diakinesis, this particular structure gets broken down. So you have chromosomes like this now, with some parts of it being exchanged during the crossing over. For example, if this was originally a yellow colored chromosome and this was a red colored chromosome, what happens after crossing over and diakinesis is that some part of this red chromosome is exchanged with the yellow one and some part of the yellow one is exchanged with the red one. This is also a symbol of completion of crossing over between the two uh, homologous chromosomes. Alright. So now that they have exchanged their genetic material, they will undergo metaphase 1. In metaphase 1, what happens is the bivalent chromosomes align on the equatorial plate, as you can see over here. The bivalent chromosomes align on the equatorial plate. The microtubules from the opposite poles attach to the pair of homologous chromosomes. These are the microtubules. They have attached to the centromeric region of the homologous chromosomes via spindle fibers. So the homologous chromosomes in anaphase, the homologous chromosomes separate. This is an important statement because when you're talking about mitosis, there's no homologous chromosomes in mitosis, right? In mitosis, it is just chromosomes and the chromatids separate. I'll just show. So this is the chromosome of mitosis. In your anaphase, what happens? The chromatids get separated, right? In case of your anaphase 1 of meiosis 1, what happens is that whole chromosomes are separated. All right. So the homologous chromosomes separate while sister chromatids remain associated at their centromere. Okay, so the whole sister chromatids are separated. So the chromatids are still not broken from each other. The whole set of sister chromatids go. Alright, I hope it's understandable. If it's not clear, please mention in the comment. I will upload another video fully explaining this part only then. So the homologous chromosomes separate while sister chromatids remain associated at the centromere. In the telophase 1, the nuclear membrane and nucleolus reappear. Cytokinesis follows, and this is called dyad of cells. Although in many cases the chromosomes do undergo some dispersion, they do not reach the extremely extended state. The stage between two meiotic divisions is known as interkinesis. So this is important. So a question may come in your exams, like board exams and all. What is interkinesis? It is generally short-lived. Interkinesis followed by prophase 2, a much simpler prophase than prophase 1. Now let's do meiosis 2. Let's move to meiosis 2. In meiosis 2 again, it will start with pro, meta, na, telo. So the first stage is prophase 2. Meiosis 2 is initiated immediately after cytokinesis. So over here, what is happening? This is called cytokinesis, right? Breaking division of cytoplasm. So this is cytokinesis. immediately after cytokinesis, usually before the chromosomes are fully elongated. In contrast to meiosis 1, meiosis 2 resembles mitosis. So meiosis 2 resembles your uh, meiosis 2 resembles your mitosis. The nuclear membrane disappears by the end of prophase 2. The chromosomes again become compact. They become compact because again now division has to occur. So that each cell gets only one chromatid instead of a pair of sister chromatids, right? Let's move ahead. So what happens in metaphase 2? At this stage, the chromosomes align at the equator. Normal metaphase behavior, right? That the chromosomes will get aligned at the equator, and the microtubules from opposite poles of the spindles get attached to the kinetochores of sister chromatids. So over here is the kinetochore. So the spindle fibers get attached to the kinetochore that is present at the centromeric region. In anaphase, there is splitting of the centromere of each chromosome, allowing them to move towards opposite cell. So what happens is that, yeah. So what happens is that since it has bound to the kinetochore at the centromere, the spindle fibers will start pulling each chromatid towards itself. So the chromatids will now get separated, right? And the separated sister chromatids will now be moving towards the opposite poles, right? So over here, as you can see, this is one chromatid, right? So in telophase 2, meiosis ends over here, in which two groups of chromosomes once again get enclosed by a nuclear envelope, and cytokinesis follows, right? So as you can see over here, we have your n number of chromatids, right? N number of chromatids, or n number of chromosomes in this case, because they are not in duplicate. I mean, what I'm trying to say is that you initially started with 2n and now you have n number of chromosomes. So what is the significance of meiosis? So maximum a 2 marks question can come from this part in your boards. So meiosis is the mechanism by which conservation of specific chromosome number of each species is achieved across generations in sexually reproducing organisms, even though the process results in reduction of chromosome number by half. So for the continuation of species, meiosis is required. Otherwise, if there was only mitosis, then your gametes would also have 2n. Your gametes would also have 2n. After fertilization, it would give you 4n. So it would result in multi-nucleated species, and the conservation of species would not have occurred. It also increases, so this was your first point, it also increases your genetic variability because of the process of when we were studying crossing over. So what happens when the non-sister chromatids cross over, they exchange the genetic information that is present between the two sister chromatids, right? So for example, from this pair some genetic information is transferred to this pair, and from this pair some is exchanged. Basically, it's not transferred, it's exchanged. The regions that come in close contact with each other exchange their genetic information. Genetic information in turn is, as you may know, it is encoded by your genetic sequence, right? So for example, instead of AGTCA, it now becomes ACTAG. Okay, so something like that. It also increases genetic variability in the population from one generation to the next. Variations are important for the process of evolution. Now, if there is no variation, all of us would look alike. And if there is, for example, a case of any disease, all of us would be possessing that disease. So wherever there is variations, it generally acts in the positive side of evolution, and we retain better traits, but it is only possible when there is a mix up of everything, right? So these are some of the important notes that I've gotten from different sources, because these sort of questions come a lot in the exam, as you will see when we will be discussing the previous year questions. So this is the first thing. So what is prophase? Prophase is the initial stage of mitosis where the chromosomes condense and be. So this is, mark the keywords. The chromosomes condense and become visible. However, the spindle fibers are not attached to kinetochores. Spindle fibers begin to form, but their attachment happens later. Their attachment basically happens in the metaphase. Now let's come to metaphase. This is the stage where the spindle fibers attach to the kinetochores of chromosome. Now again, what is kinetochore? If this is your chromosome, they are attached at the centromere. On the centromere is present a proteinaceous case which is known as kinetochore. Okay, so this particular structure is your kinetochore. Kinetochore. During metaphase, chromosomes align at the metaphase plate, also known as equatorial plane. This alignment is facilitated by the spindle fibers, right? Over here, spindle fibers are attached. Over here, spindle fibers are attached. Accurate separation into daughter cells. Then comes anaphase. During anaphase, the sister chromatids separate in this fashion. So, and are pulled apart as the spindle fibers shorten. While spindle fibers are essential during this phase for pulling apart the chromatids, their initial attachment to kinetochores occurs earlier. Then comes telophase. This is the final stage of mitosis where the chromosomes reach the opposite poles of the cell and the cell starts to reform its nuclear membrane. Spindle fibers disassemble during this stage. In oocytes of some vertebrates, diplotin lasts for months. Yeah, this was again an important statement. I have also mentioned it while teaching you the statement, but just for your understanding, this is an important statement, so mark it. Yeah, so now let's move into different phases of prophase 1. A lot of questions have come from this part, as you will see, and we'll be discussing the questions. But in general, also, it's very important for you to know. So the first stage is leptotin. As we've discussed, what happens in leptotin is that the chromosomes become visible. So this is the first stage when chromosomes start to become visible under the microscope as thin thread-like structures. Hence, it is characterized by chromosomes looking like thin threads in case of meiosis 1. Then let's talk about zygotine. During this stage, synapsis begins. So this is the keyword, synapsis begins, which is the pairing of homologous chromosomes. So what is synapsis? Synapsis is basically your pairing of homologous chromosomes to form bivalents, right? So if this is your first chromosome, this is your second chromosome, these are your homologous chromosomes, these will form a bivalent. And this whole process of formation of bivalent is known as synapsis. This corresponds to the formation of the synaptonemal complex. Now we'll talk about pachytene. At this stage, crossing over occurs. So crossing over is the key word, where genetic material is exchanged between the homologous chromosomes. Crossing over looks something like this, right? So this part and this part, they are undergoing crossing over. Appearance of recombination nodules. This is also known as recombination nodules, which are essential for crossing over, characterize this stage. Now let's move to dikinesis. Dikinesis is the final stage of prophase 1 where chiasmata move to the end of the chromosome, a process known as terminalization. So whenever there is crossing over, this particular part is known as your chiasmata. That is the site of crossing over. So this chiasmata is also characterized by the presence of a proteinaceous ring around it, so that the homologous chromosomes find it easier to undergo recombination. So what happens in dikinesis is that this chiasmata is terminated so that now the homologous chromosomes can separate from each other, right? Hence, this phase is marked by the completion of terminalization of the chiasmata. So, this is very, very, very important. When we are talking about synaptonemal complex formation, it will be zygotine. When we are talking about terminalization of chiasmata, it will be dikinesis. When we are talking about chromosomes looking like thin threads or visualization of chromosomes in meiosis, it will be leptotin. That is the first stage. And then when we are talking about recombination nodules, it is pachytene. All right. This is the end slide. And before I end this, I would like to first tell you a few things. So when we are talking about mitosis, and I have already explained this before as well, G1, S, G2, and then your mitosis starts. So if you have initial number of chromosomes and this is your DNA, okay? So if your initial number of chromosomes is 2n, then the amount of genetic content in your G1 would also be 2n. In your S phase, the number of chromosomes remains 2n, but the DNA becomes 4n because in S phase, we have the process of replication. DNA replication occurs in S phase. Alright. In G2 phase, only proteins are synthesized. More and more proteins are synthesized so that the cell finds it easier to divide. So over here, chromosomes will be 2n and DNA will again be 4n because there has been no increase or decrease after the replication process. However, after mitosis, what happens? The number of chromosomes become 2n and DNA also becomes 2n in each daughter cell. Daughter cell. Now let's talk about meiosis, and specifically meiosis 1 at this stage. So in meiosis 1, again we'll draw chromosomes and the genetic content or DNA. The first stage is prophase, pro, meta, ana, and telophase. But before this, S phase has already occurred. So the number of chromosomes is 2n, but the genetic content is 4n because of S phase. Then comes pro, meta, ana, and telo. So in your pro phase, what happens is that the chromosomes are still 2n and DNA is 4n because still prophase, no division, nothing has happened. In metaphase also, it's 2n and 4n because in metaphase, if you remember, what happens is that the homologous chromosomes are now at the equatorial plane, but till now no cytokinesis has occurred. In anaphase, what happens is that they start getting pulled towards the poles, right? They start getting pulled towards the poles, but still there has been no cytokinesis. So the number of chromosomes is 2n and the DNA content is still 4n. When we're talking about telophase, after telophase cytokinesis occurs. So over here also is 2n and 4n because even though they are at separate poles, they are completely at opposite poles, but till now cytokinesis has not happened. Since cytokinesis has not happened in one cell, the total amount is still 2n and 4n. After telophase is completed, cytokinesis occurs, right? So at the end of cytokinesis, each daughter cell would now have, since 2n is getting divided between two parts, so each daughter cell would now have n number of chromosomes, but genetic content would be 2n, right? So this is, for example, imagine that the cell has only one pair of one pair of chromosomes. So 2n means 2, right? If n is 1, then 2n, that is the diploid content, means 2, like sort of this scenario. So over here, these are your two pairs of chromosomes. Now, when these are getting separate, sorry, two pairs of chromatids, one pair of chromosomes. So if the chromosomes are separated, so one pair of chromatids go to this side, but it's one chromosome, right? And the other chromosome goes to this cell. So each daughter cell now would have n number of chromosomes. So this is the situation of in each daughter cell. Now till this was the part about your meiosis 1. Let's move to meiosis 2 now. Let me just quickly rub off so that I can make some space. Yeah, so let's move to meiosis 2. Now since cytoplasmic division has already occurred, cytokinesis has already occurred, so now what we are left with is that each cell has n number of chromosomes and the DNA content is 2n. And meiosis 2 also looks like mitosis. That means equational division. Meiosis 1 is reductional division. So you start with 2n, it gets reduced to n. So meiosis 1 is reductional, reductional. However, meiosis 2 is equational. That means if each cell is starting with n number of chromosomes, it will end with n number of chromosomes only. So it's equal. From the beginning to the end, the number of chromosomes remain as n. So this is your starting point. Start with n and 2n. Now we'll move to prophase 2, pro 2, pro, meta, ana, telo. 2, 2, 2. Right. So the number of chromosomes will remain as n. In case of your metaphase, what happens is, yeah. So what each cell would be looking like would be something like this. In prophase, it would look like this. Each cell will be having one chromosome to maintain the analogy. In metaphase, this pair of chromatids or this one chromosome will arrange itself at the equatorial plane. In anaphase 2, what happens is it gets separated. Now, if you're looking at it from an outside point of view, it would look like you have two different chromatids. You have the sister chromatids are separated. So now you have two chromatids. So it would look like 2n, right? The DNA content, however, remains constant 2n, 2n, and 2n because even though the chromosomes look like there are 2n chromosomes, but the genetic content is 2n only because no replication or anything has occurred. The genetic content remains constant. In case of your telophase 2, what happens? Now in telophase 2, they are completely at opposite poles, and telophase 2 plus cytokinesis. So at the end of your cytokinesis, what happens is the furrow has formed and the cells have divided, and each cell now has, so 2n gets divided into two cells, so each cell has n number of chromosomes, n chromosomes, and similarly the total genetic content is also divided between the two cells, so each cell will have n amount of genetic content. So if we talk about meiosis from the beginning to the end, what happens is your 2n amount of chromosomes get converted to n amount of chromosomes, with each cell having n amount of genetic content as well. Alright, I hope it was clear. Now let's just, I will also summarize your mitosis in terms of your chromosomes and DNA content. Till now, in terms of mitosis, we have talked about G1 phase, S phase, and G2 phase. However, there are multiple other phases of mitosis. So, when we're talking about mitosis, G1, S, G2, and then pro, meta, ana, telo. This is chromosome and this is DNA. So the number of chromosomes you start with is 2n. DNA is also 2n. In S phase it becomes 2n, it becomes 4n. In G2 phase it becomes 2n. This is also 4n. Right. Till this part we have already discussed before. Now from pro, meta, ana and telo, we'll move on. So in prophase, the only thing that happens is that the chromosomes become condensed and visible. So nothing of separation occurs. So it's the same till this part. Then in metaphase, what happens is that all of the chromosomes are aligned at the equatorial plane. So again, no division or anything has occurred. So it's 2n and 4n. Then we'll talk about anaphase. In anaphase, what happens is that the sister chromatids get separated, right? So this is and this. Sorry, I have become so used to making an X that I always make an X. Anyway, this, then this, then this, then this, right? Because I've drawn three chromosomes, so I'm going to continue with that. So you can see over here, if you look at the structures are three. If you look over here, the structures have become six. This happens in anaphase. So in anaphase, the number of chromosomes becomes 4n. If n was 3, so initially we were saying 3. When we reach anaphase, it becomes 6, right? So in anaphase, the number of chromosomes also gets doubled because there is a splitting from the center, that is, there is a splitting from the kinetochore, and the sister chromatids get separated, right? However, the genetic content is still 4n because no replication has occurred. Genetic content will only change whenever there is replication. Replication only occurs at the S phase. So once in the S phase 4n has formed, especially in case of mitosis, it will remain 4n till the end, after which, after cytokinesis, you will get 2n of DNA in each cell. Alright. So in telophase plus cytokinesis, what happens is that each of these start getting accumulated in one cell. So the number of chromosomes in each cell is now 2n, because we were starting with 2n equals to 3. 2n equals to 3 and ending with 2n equals to 6 at anaphase. Now again this gets separated. So in telophase you will get 2n equals to 3, because over here 2n is equals to 3. This is one cell, 2n is equals to 3, this is another cell. So in telophase you will get 2n, and the amount of DNA is also now separated between the two cells, so the amount of DNA would become 2n. Okay, so questions have not come in neat from this part per se, the last slide which I have taught, but it was an important slide because it is important for you to get your concepts cleared. In case there was any difficulty in understanding any of the statements that I said, you can reach out to me. Alright, I will also give my email ID in the description. So if you want, you can reach out to me personally, if not on the comment box. However, I would encourage you to use the comment box so that whatever problem you have can also, if I solve it publicly, then others may also benefit from it. So this was pretty much it from my side. If you all have any questions, feel free to ask. We'll be discussing the previous 10 years questions in your next video. And okay, goodbye till then. Hopefully I'll be releasing it all soon. Oh yeah, once more, like, share and subscribe.